Dear participants, my name is Oksana Bahrizayets. I would like to present the result of our research on the topic mathematical and computer simulation of the response of potentiometric biosensor for the determination of alpha-chaconine. Nowadays, the concept of creating cyber-physical systems for various spheres of technology interaction with human activity is actively developing. Our work was supported by Erasmus project. The future is in applied artificial intelligence. In general, cyber-physical systems are considered as intelligent systems in which external devices, processors, physical objects, network equipment are integrated. The main purpose of cyber-physical systems creation is to monitor the behavior of physical objects as components of such systems in real time. These are systems in which cybernetic means measuring, computing, control interact with physical processes in arbitrary objects. Cyber-physical systems are attracting more attention than ever thanks to the rapid development of the Internet of Things and its combination with artificial intelligence, machine learning, and the cloud. Cyber-physics of the system are identified with the manifestation of fourth industrial revolution that is taking place in the modern world. Thus, there is also the physical possibility of using Internet of Things technologies where signals from sensors and measuring instruments need to be used. Cyber-physical system technologies are already transforming the way people interact with engineered systems in the real or physical world, just as the Internet has transformed the way people interact with information. Yet, due to their complexity, the developers of cyber-physical systems face a major problem: the lack of simulation tools and models for their design and analysis. Cyber-physical systems are systems that integrate computing elements with the physical components and processes. The computing elements coordinate and communicate with sensors, which monitor cyber and physical indicators, and actuators, which modify the cyber and physical environment. Biosensors are analytical devices that use biochemical reactions to detect a wide range of chemical compounds, metabolites, medications, environmental pollutants, etc. This slide shows the principle of operation of cyber-physical biosensor systems, which can be used for medical and biological processes. The first, a particle of substance is measured, resulting in the formation of the product, which is converted by the converter into an electrical signal for data analysis. That is, the methods of molecular biology, electronics, and communications make it possible to monitor health, controlling chronic and preventing critical human conditions. Nowadays, there are different types of biosensors depending on the type of transducer, bioreceptor, and immobilization. Biosensors measure biological or chemical reactions by generating signals proportional to the concentration of an analyte in the reaction. The biosensor itself is typically made up of the following components. First, it's the analyte. This is the substance that is being detected. The bioreceptor is a molecule that recognizes this analyte. Examples of biosensors inside the body include enzymes, cells, and antibodies. The process of signal generation upon interaction with the bioreceptor with the analyte is called biorecognition. Next is a transducer is the element converting the forms of energy through the process which is called signalization. In this case, it's used to convert the biorecognition event into the measurable signal. The electronics element processes this transduced signal and prepares it for quantifying on the display. Finally, the display consists of a user interpretation system similar to that of the mobile phone screen. This part consists of hardware and software that will display the generated results in the user-friendly way. The display may show a numerical value and image or data in the table graphical form, depending on the requirements. In our work, we are considering alpha-chaconine. Alpha-chaconine is a steroidal glycoalkaloid. It's natural toxicant produced in green potatoes and gives the potato a bitter taste. The question is extremely relevant given that alpha-chaconine is a very interesting object from a biological point of view due to its toxicity and the determination of its concentration in potatoes as a very popular food product, due to which potatoes acquire a bitter taste. Measurement of the content of alpha-chaconine in potatoes is carried out when new varieties with reduced content are bred. The currently developed methods for determination of the alpha-chaconine are based on the colorimetry, chromatography methods, and radio or immunological analysis. But these methods are characterized with a very high cost, long duration, and complexity of simple preparation methods. In order to optimize and modify the existing methods, it's recommended to use more cheaper, more simple, and also a highly sensitive method, which is based on the using of biosensors. At the same time, with the purpose to save the time and to save materials, it's economically beneficial to create and research adequate mathematical models of biosensors for determination of alpha-chaconine with the possibility of varying the simulated response. The structure of ion-sensitive field effect transistor is shown in the slide. It consists of the source, drain, gate insulator, and reference electrode. The amount of the current flow will be not only determined by the change of the biomolecules interacting on the gate dielectric, but also sensitive to pH, different ions, products of enzyme reaction, etc. In our work, we are developing a mathematical model of the previously developed butyrylcholine esterase biosensor, which is based on the ion selective field effect transistor for the inhibitory determination of alpha-chaconine. In recent years, scientific research has been conducted based on the results of which it can be concluded that mechanisms of resistance of potatoes to diseases and effects of insects depend on the level of alpha-chaconine. Among other factors that affect the level of alpha-chaconine, it can cause a significant increase in its initial concentration. It should be noted climatic changes, the effect of light, mechanical damage, and measurements which bring us constants were carried out using the portable device which was developed and manufactured at the Institute of Semiconductor Physics of National Academy of Science of Ukraine. In the slide, we're able to see the schematic representation of the operation of the biosensor, which is used for measuring the alpha-chaconine. Actually, this biosensor consists of 3 stages. obtaining a baseline stage 0 response to the working concentration as a substance stage number 1, and the response to alpha-chaconine as inhibitor stage 2. The functioning of the biosensor is based on enzymatic reaction that takes place in the bioselective membrane and can be presented in the following equation, which is shown in the slide. During the chemical reaction, protons are generated, which leads to a change in the pH inside the membrane. So it's advisable to use potentiometric biosensor, which is based on the pH-sensitive field effect transistor. Now we're able to see the schematic representation of the enzymatic reaction, and the mechanism of inhibition by alpha-chaconine belongs to the mixed type of inhibition. And this scheme we're able to see. Here we have the enzyme, substrate, and inhibitor. Also, all the components are listed down, and biochemical reaction rate constants K are difficult to obtain directly from experiment. In our work, these constants were selected in such a way that the model response coincided with the experimental responses. We built the system of 7 differential equations, and here we have the concentrations of enzyme, substrate, inhibitor, product, and also complexes: enzyme substrate, enzyme inhibitor, enzyme substrate inhibitor. And the change in the product concentration over time is directly proportional to the response of the biosensor. Also, we have constants which are in the system of 7 differential equations. All initial concentrations are obtained from the experimental data. And this experimental response was obtained at the concentration of an enzyme 5.8 times 10 to power minus 4 mole per liter. Experimental response of potentiometric biosensor for determination of alpha-chaconine we're able to see in the slide. And according to these results, the response of the biosensor for determination of alpha-chaconine was obtained at concentrations which are shown in the slide. And analyzing the numerical simulation results, we may summarize that the higher the concentration of the inhibitor, the smaller the response amplitude of the biosensor model under study. The simulated reactions of the biosensor at different concentrations of inhibitor fully corresponds to the principle of inhibition. And finally, we compared experimental data and model results. Model curve is in red and experimental is in black. And we may summarize that the input parameters of the system, their selection was carried out in such a way that the simulated response of the studied biosensor coincided with the experimental one on as much as possible. And about the absolute error of the simulation, the maximum verification error is manifested in the area of stabilization of the response of biosensor to the action of the inhibition and doesn't exceed 0.045 count units. And finally, the use of mathematical modeling to optimize the analytical characteristics of biosensor for determination of alpha-chaconine will allow to minimize laboratory experiments with toxic and expensive substances in order to select optimal concentrations of components. In future research, it's necessary to investigate the states of equilibrium and stability of developed mathematical models to develop software complex and design of cyber-physical systems for such biological processes. Thank you very much for your attention.